Everyone, my name is Shagun, as you guys already know, and today I'm going to be presenting a paper that I found really interesting, and it's called Extracting a Biologically Relevant Latent Space from Cancer Transcriptomes using a Variational Autoencoder. So that's a bunch of words, but essentially what this means is using a Variational Autoencoder to identify biological patterns within the data and also being able to reconstruct the data. And the data set is partaken from the Cancer Genome Atlas. So it's this huge data set profiling over 10,000 tumors, over 33 different cancer types.
The clicker.
So most of you probably know what an autoencoder is, but there's a stark difference between an autoencoder and a variational autoencoder. And that's because an autoencoder is deterministic. So whatever the output of the autoencoder is solely based on the parameters and the initial coding of whatever the data through the autoencoder is. So as you can see, this is an autoencoder. So whatever data you put in the input layer will then be compressed into a hidden layer, and then will be then reconstructed in the output layer. But then also, when the data is compressed, it's only outputted as a single. It's only encoded as a single value. Now, if you look at a variational autoencoder, when it's compressed, when the data is compressed from the encoder, it's represented as a probability distribution. So if you look at a variational autoencoder, obviously it looks more complicated and it has more features. So essentially, a variational autoencoder is compressed of three fundamental parts: an encoder network, a decoder network, and a loss function. So the encoder network basically takes the data and encodes it, and when it encodes the data, it compresses it, and it compresses it into a number of different features. But it represents this data as a probability distribution. Now this probability distribution is forward propagated into two vectors: the standard deviation vector and a mean vector. So the standard deviation vector basically tells us the area from where the encoding or the probability distribution can vary from the mean. And then the mean vector basically controls the range. So the range from where the data or the encoding can be central around. From then, the decoder network comes into play, because the decoder network will then sample from these two vectors to get a vector of features to forward propagate into the decoder network to reconstruct the input data, and basically try to get the output as similar to the input in the encoder network. So the loss function is basically comprised of two parts: the generative loss and the latent loss. So the generative loss is your standard mean square difference between the output of the decoder network and the input of the encoder network. But then the latent loss is actually the difference between two probability distributions. And the two probability distributions is, if you look back, is the probability distribution between the sampling from the decoder network and the probability distribution from the output of the encoder network. And how to improve the model is to backpropagate it, backpropagate the gradient to adjust the parameters and the weights for these two losses. So in the paper, the data consisted of 5,000 variably expressed genes. And so basically it was the genes and their gene expression level. But these genes were stemmed from, as I mentioned before, the Cancer Genome Atlas. So all of these genes were taken from a different series of cancer's tumors. And what's really interesting is, most of you might not know this, but a gene expression level is basically a measurement from 0 to 1, in this paper specifically, of how active or inactive a gene is. So if a gene is active, that means its gene process is being carried out. So essentially, when a gene is active, it encodes its DNA into an RNA called transcription, and then from an RNA to a protein called translation. And what happens is, this is a huge critical process, because basically a protein dictates cellular function. So if any malfunction or dysfunction happens between gene expression or the gene process, then this could cause something like cancerous tumors. So the main purpose for this paper and the main purpose for this model was to actually extract and understand if the variational autoencoder could learn an underlying manifold of the data and could also construct and identify patterns within the data. So in order to see if the variational autoencoder could identify patterns within the data, what the researchers did was that they actually extracted the weights from the layers of the decoder model. And the reason why they did this is because essentially when the decoder network is sampling from the two vectors of the encoder network, it learns an underlying distribution of the data. It also understands and learns which genes contribute to which specific features that are encoded within the latent space of the encoder network. So from extracting the weights in the decoder network, the researchers were able to identify if the variational autoencoder was able to identify features within the data set. So in fact it did. So it was able to identify and robustly separate patient sex and which genes contributed to patient sex and create a constructed feature out of it. So when extracting the weights from the decoder network, in feature 82, you can see that all the genes compressed in feature 82 were either found on the X chromosome or the Y chromosome. And yeah.
Sorry, I'm a little confused. Probably said it, but I didn't catch it quite. Usually, when you go from your initial space to the representation space, the vectors in that space don't really mean anything. But here, you're saying that one of the features, for example, means the patient's sex. So, is that by design?
Yes.
And how do they get that to be by design like that?
So basically, the data set that was encoded within the variational autoencoder by the encoder network, it was split into, I didn't mention this, it was the 5,000 genes variably expressed as the data set to be encoded, and then the encoder network constructed 100 features. So from those 100 features, it was able to identify that these genes have similar characteristics and could be grouped together in a feature. Did that answer it?
Clustering? Did they run a clustering algorithm? How did you put these genes together?
I'm actually not sure.
So isn't it what the autoencoder is doing for you?
Yeah, that's what I was saying.
So the autoencoder just encodes the representation, right?
Right, but
you can if you have these features, if you know these features as labels in your data set, you can either do correlation analysis and see what components of the encoded vectors relate to what features in the data set, or alternatively, you can just use a clustering algorithm to cluster your data and see which cluster relates to what.
But for example, if you have a bunch of different genes that result in the first or the 82nd feature to be prominent, isn't that sort of clustering?
I see. So you're talking, basically what you're saying is that one of my features correlates with something in my data set, which is the same thing.
Right.
So yes, you can look at the components, see if it fires when there is some feature in my data set present. Does that make sense?
Yes.
Oh, you were inside. I thought you were distant.
It really handles like the
Do you want to come sit here so that you're closer to the camera?
But one problem happens when there are interactions. When interactions happen, which I think in the genomes we do have interactions, there are algorithms called disentanglement algorithms, which take care of those interactions to some extent, not perfectly. Yeah, but
Awesome. Let me see that the paper's out. Good to know. So it was also able to identify the features that separated primary and metastatic skin-cutaneous melanoma tumors. So as you can see in feature 66, it identified which genes contributed to the metastatic skin-cutaneous melanoma tumors and which genes contributed to the primary skin-cutaneous melanoma tumors. Now, something called overrepresentation analysis was actually done on these features. So overrepresentation analysis is a technique to identify Go terms or gene ontology terms in a gene set, and for this specifically feature 66. So overrepresentation analysis was able to identify Go terms in gene 66. So Go terms can be characterized in 3 sets. So they can be molecular functions, they can be biological processes, and they can also be I forgot the last one. But basically, from feature 66, they were able to identify the Go terms relating to cholesterol, lipid metabolism, and also ethanol. And what this means is that basically doing overrepresentation analysis over this gene set, you're able to find the biological process that's overrepresented between these genes. So overrepresentation analysis gives the Go terms for each gene in the gene set. Then they look at which Go term is overrepresented within that gene set. So whatever Go term is overrepresented within that gene set, that means it's not random and actually has a strong correlation, which can actually help researchers identify if they should focus on that specifically to see if that particular pathway or that those genes relating to cholesterol can have an effect in causing the cancerous tumors. So again, this can help researchers identify and create drugs to specifically target areas which can cause cancer by just understanding the Go terms of a particular gene set or feature 66. So some of the key takeaways is that a variational autoencoder is a generative model, so it's able to reconstruct the data, but it's also able to understand the underlying representation of the data. So while also, as you said, it could be a clustering algorithm that was able to identify was able to create and construct the different features within the data, and the different features related to patient sex, and they also related to identifying and separating primary and metastatic skin cutaneous melanoma tumors.
Can I stop you? Not only because you looked at it, but also I have this question for a while. I think.
Sorry, can you speak up a little bit?
Yeah, sure. Thanks for saying.
Is the microphone working actually?
The microphone is doing something else. It's not for you. But
we might have a microphone as part of this whole setup. In case you want to use it.
Yeah, do you want to use it now?
Just keep going, I'll figure it out.
So we'll think about it. But meanwhile, we speak louder. Is it loud enough? Okay. So basically, my question is: you did mention, just to figure out that part, I'm still sort of struggling. You did mention that you were recording the weights of your decoder. How does that relate to determining the sex of the patient?
Yeah, so basically the weights of the decoder model are able to capture which genes contribute to which feature. So when you see which genes contribute to which feature, you were able to see in feature, I think it was feature 53, you're able to see that all the genes in that feature are found in the X chromosome or the Y chromosome. So then you understand that the variational autoencoder was able to understand that there was a biological variance within the data.
Yeah.
And then you're able to see that, oh yeah, I identified that there's patient sex.
So you are looking at the correlation between the representation of your, the components of your representation and the feature that you know in your data set, in this case, is the sex. So I just wanted to figure that out.
I have one question. So this one, you know that there are male, female, and these features. So is there a way that you come up with something which is unknown, a relationship which is unknown?
So actually, let me go back. Encoding 53, when overrepresentation analysis was on encoding 53, there was actually no Go terms related to that. So there was an unknown biological component that could be related to the genes encoded in feature 53. So even after trying to identify if there was an overrepresented Go term within this gene set, none was found. So that just tells us that there's an unknown biological process within this gene set. So yeah. So I'm guessing we do discussion questions after, or yeah. Or
The
you can wrap.
summary slide. Yeah.
Or do you want me to explain this again?
Sure.
So
Let's give her a
hold
round.
on. What we're going to do is that we're going to take a break now, and then we start from the takeaways and then go into discussion.
Let's give her a round of applause.